Good morning, everybody. So this lecture is going to be about the genome-based taxonomy, and this is part of the Microbiome Informatics webinar series, which is being hosted by the Ohio State University. So we are now on week 7 here. So just to introduce myself, I'm Donovan Parks. I'm a computer engineer and computer scientist by training. I did my undergrad at the University of Victoria. It's in British Columbia, Canada. Then did a master's degree in computer vision at McGill. And it's at that point I switched over to sort of microbiology, though I did a PhD out of the computer science department at Dalhousie University, but my supervisor was Rob Biko, who is a trained microbiologist. And my PhD was basically on microbial biogeography. I then did postdoc work in Australia with Gene Tyson and Phil Hugenholtz at the University of Queensland, and that's where I got into sort of genome-based community analysis, in particular metagenome assembled genomes, or MAGs, and eventually got involved with the Genome Taxonomy database, which will be the last half of this lecture today. So what I'm going to go over today is just a few different topics. I'm going to sort of introduce some of the more fundamental concepts of nomenclature and taxonomy, then give a brief introduction to species concepts, and then focus more on species definition. So that's how, in a practical sense, can we assign organisms or strains to species? What are people in the field currently doing in order to address that problem? So that'll probably take about the first hour of this lecture. And then in the last half, I'll discuss the Genome Taxonomy database, explain basically how that works, and use that as an example of how you can apply these principles of nomenclature and taxonomy to a large taxonomic framework. And then I'll just walk you through the actual GTB website to show you what kind of information is available there, and through GTB-TK, which is basically a companion tool which allows you to submit your genomes, whether those be isolates, MAGs, or SEGs, and get back a taxonomic classification. Now, I wanted to start just by sort of setting the table. So I think very reasonably, there's a perception that nomenclature and taxonomy are sort of solved problems, that there are just names that we have for different bacterial and archaeal groups, and these are sort of set in stone. That was certainly my perception before I started becoming involved in these fields, but really that couldn't be sort of further from the truth. And certainly today, these are actually very topical areas of study, and there is sort of different opinions in our community about how we should move forward with nomenclature and taxonomy. So I just wanted to walk you through this slide just to give you sort of a brief history of sort of where we are in 2021, which I think will help give you a better context for the first half of this lecture, where we're going to go over some of the fundamental concepts so you can kind of understand and appreciate how those sort of fit into modern day studies and what people are thinking. So if we take you back sort of the early days here, which for the purposes of this talk is pretty much before 2010, so not that long ago, we have a code of nomenclature, which we call the Prokaryotic code, which we'll discuss in some detail here. And we basically have taxonomic opinion in our community, which basically gets codified in the NCBI taxonomy. So it's basically their mandate to look across the published literature in our community and decide what the current taxonomic opinion is. And arguably this rule of nomenclature and taxonomy was serving our community very well, and many will argue that it still is. And I think largely it worked because the rate at which we were discovering new taxa, and in particular new species, was slow enough that these systems could keep pace. Now this sort of gets contrasted with where we are today. So let's take you over to say 2010 to present day, where the cost of sequencing has gone down a lot. This is allowing us to get isolate genomes at scale. So we have initiatives such as GIBA, where they're systematically going through and trying to sequence every type strain of a species. Again, if you don't know what that is, I'll explain later. But the upshot of that is they're coming out with papers where they have 1,000 new isolate genomes in one paper. And even probably more salient to this question here is, of course, now we have these metagenome assembled genomes, or MAGs, where we can pull out genomes directly from environmental samples. And there are now papers coming out where we're literally pulling out 10,000 or more MAGs in a sort of single study. And these represent hundreds, if not thousands, of new species. And these are really stressing these sort of traditional methods for doing nomenclature and taxonomy. So if I take you to 2021 here, so where we are, the upshot of this is now we have the prokaryotic code, which we've been using for, I guess, about 6 decades now. And there are people now in our community that are suggesting this code is not keeping pace with this new discovery, that it actually represents a bottleneck. So there are efforts now to propose a new nomenclature code called SEEK code. And similarly, we have the NCBI taxonomy, which sort of operates at the pace at which we can publish new peer-reviewed literature. And we have the GTDB, which sort of takes the view that we are discovering new taxa at such a rapid rate that we need a more systematic way to do taxonomy, and one that sort of has more quantitative rules and a framework where we can sort of update it every 6 months or 8 months with all the new genomes that we're discovering. So here we are in 2021, where we basically now have 2 essentially competing codes of nomenclature, at least 2 sort of commonly used taxonomic frameworks. And this is really being driven by the pace of new discovery, in particular, the number of MAGs we're able to pull out to sort of have genomic representative of this sort of uncultivated majority, which we've been talking about for the last couple of decades. Okay, so with that sort of setting of the table, I just want to take us back sort of the very basics here, just to make sure we're all on the same page. So a taxon, in its most simplest definition, is just a group of organisms, and the plural of taxon is a taxa. So I'm going to be using those terms a lot in this lecture. Now, generally, when we talk about taxon these days, we mean something that's in a hierarchical system. So I'm sure you've all seen this, where we have taxa going from species to genus up to domain. And this really dates back to Linnaeus in the 1700s. And it is just simply proven to be a useful way to describe biological diversity. So we continue to use it. Now, we also generally mean for a taxa to be a group of organisms that are evolutionarily related. And this dates back to Darwin and this concept that we have descent with modification. And I would say most people today, when they are describing a taxon, what they really mean is something that is strictly monophyletic in some modern inference of the evolutionary relationships between those organisms. Okay, so there are sort of 2 related concepts that I want to go over with you. So the first is basically nomenclature, which tries to answer the question, how do we propose and assign names to taxa? And taxonomy, which addresses the question of how do we decide which organisms or strains actually belong to a taxa? So this little sort of cartoon illustration at the bottom here sort of tries to depict what these 2 concepts are and how they are related but different. So what I'm showing you here is 10 different strains shown by these little cartoon bacterium. And these boxes are different ways which we might decide that these strains belong to a species. So you can imagine we might decide that these 5 strains are a species, or we might decide that these 8 strains are a species, or that actually all 10 of these strains are a species. So that is the question of taxonomy. How do we decide how to draw this box? Which strains do we want to say belong to a species? Nomenclature, on the other hand, is concerned with, once we've decided what the box is, how do we actually give a name to this box? How do we give a name to this box? And as you'll see, there are basically a set of rules for this. And the purpose here is to have some sort of organized framework so that me and you can communicate effectively about the taxa we're talking about. So in the sense that we will agree on a set of names. Okay, so by far the most important thing to understand about nomenclature is that there are a set of rules and that the community as a whole that we work with are following these rules. And these rules are put forth in what's called the International Code of Nomenclature of Prokaryotes, People typically just refer to this as the code or the prokaryotic code, and I will do the same here. And this code is governed by a committee. So this is just a group of, I think it's about 8 people in our community that form the International Committee on Systematics of Prokaryotes, or ICSP. So the URL at the bottom here is a link to this actual code. Of course, it's a written document. It's about, I think it's like 110 pages long or so. I am certainly not going to go through all that here. What I will do, though, is take you through, I think, some of the more salient points in the code and the sort of questions that I think you're likely to face in your own research, or that are at least useful to understand so that you can appreciate where names for taxa come from and why they may differ between different taxonomic frameworks. So if you go to the NCBI or GTB, or if you're working with 16S, maybe you're going to Silva, and perhaps the names don't agree, this will give you some sense of where these names come from and why there can be sort of a different opinion in that regard. So the most sort of important point in the code is that every name is anchored by type material. And that's what's shown in this sort of diagram in the center here. And I think it's probably easiest just to understand this by going through it with an example. So what I'm showing at the bottom here is a strain. So this is something that we actually have cultivated. And the idea here is this strain is an actual biological entity. This is something that exists in nature. So ultimately, all the names we should use should tie back to this strain because it's the real biological thing that we care about. And what this flowchart is showing is that the type of a species is a strain. So in particular, just to give you a concrete example, we can have the strain Bacillus subtilis ATCC6051, and that is considered to be the type strain of the species Bacillus subtilis. And you can see that the name of this species, of course, derives directly from the strain. So in a similar way, if we look at a family, we say that the type of a family is a genus, and you get Bacilliaceae, which is, of course, deriving directly from the name Bacillus. Now, one interesting aspect of the code is that it actually has only recently covered the rank of phylum, actually in late 2020. So going back to our example on the right here, you can see that the phylum here is actually Firmicutes. That obviously doesn't follow the general naming convention here. So if this is going to get updated to follow the code, it should arguably be named Bacilliota. Now, there is a sort of discretionary room in the code where the committee is allowed to decide if there should be cases where there can be exceptions to the code. Now, this might be one of those cases, so this will essentially come down to a vote by the committee of whether or not we need to update the name Firmicutes to Bacilliota to strictly follow the code, or whether or not Firmicutes is in such common use and so widely used in our community that it just makes sense to retain it. All right, so I just wanted to sort of walk you through some examples of how you apply the nomenclatural code in practice. So what I'm showing you here is basically if we have a family and it encompasses all these genera, how do we decide what we actually name this family? And this is relatively straightforward. So first we have to decide, well, we have to look up the fact that the type of a family is a genus. So this is why we are looking at these genera. And then all names in the nomenclatural code basically have a date of priority associated with them. And this is basically the date in which this name was first published. So we look across these genera, we find the one that was published first. That's the name that has priority. It's Bacillus in this case, so we call this family Bacilliaceae. This is another example. So this is more sort of a hypothetical example. So let's imagine there is a new method that comes out for inferring, for inferring phylogenetic relationships. We believe it to be far superior to methods that we've currently been using. So we go in and we explore the phylogenetic relationship between a bunch of Bacillus strains, and lo and behold, what we find is that, oh, it turns out the strains that we were calling Bacillus subtilis are no longer monophyletic. So we have 3 strains of Bacillus subtilis at the top of the tree here, and 2 strains at the bottom here, and these 2 Bacillus agri strains between them. So the question then is, well, how we would like our taxa to be monophyletic, so how do we decide which of these 2 Bacillus subtilis lineages should retain the name Bacillus subtilis? And again, this isn't a matter of opinion. The rule basically, the code covers how you have to apply these names. So in this case, we need to, again, consult type material. So we can see that the type of Bacillus subtilis is this particular strain, which is here. So we are obliged to call this lineage Bacillus subtilis. And we need to propose a new species name for this other lineage, which we would no longer believe are actually strains that belong to Bacillus subtilis. Now, this is just one way to resolve this situation. And as I mentioned at the beginning here, deciding what strains belong to a given taxa or species is the domain of taxonomy, and taxonomy is actually just opinion. So an alternative taxonomic opinion is that all these strains actually represent a single species. So we need to now determine, well, what do we call this new species that actually encompasses strains that we previously were classifying as Bacillus subtilis and Bacillus agri. So again, we go back to type material. So that's what's highlighted in red here. So Bacillus subtilis, the type strain for that was published in 1872. The type strain for Bacillus agri was published in 1993. So Bacillus subtilis has priority. So we're going to call this species Bacillus subtilis, and Bacillus agri, because the type material is in there, becomes basically a synonym of Bacillus subtilis. All right, so hopefully that gives you a sense of sort of the fact that nomenclature does have a set of rules that need to be followed. It's based on type material. This basically allows us to determine what names should be, so that the idea being that me and you will basically agree on these names so that we can effectively communicate about these taxa in the published literature. So where do you actually get this information if you need to apply these rules? There are a few different sources, but I think by far the most commonly consulted source in our field is the list of prokaryotic names with standing in nomenclature, or LPSN. So this is a website you can go visit. What I'm showing you here is a screen grab directly from their website for Bacillus. And as you can see, it contains a lot of information. In particular, it contains the year of priority, which you're going to need to know. It indicates that the type species of the genus Bacillus is Bacillus subtilis. And it also indicates that this name is validly published. So there is a process you need to go through to get names validly published, which I'll explain in a bit. But there are entries at LPSN where things are only effectively published, or they're candidatus names, and those don't actually have any sort of priority when it comes to determining the final names of taxa. All right, so just before we sort of leave nomenclature, I wanted to go through how you actually name a new species. This is something some of you may need to do in your research. If you're doing some study and you find some strains that are novel from anything that's been described before, you're going to want to propose a new species name. We're also going to come back to this later in the talk, as this is basically, this process is the reason that some people in our community feel like we need a new code of nomenclature basically to address how this is done. So what I'm so how this works is, imagine you're doing some study, you've discovered a new strain, and you believe this is a new species, so you want to get it validly recognized. So the first thing you need to do is actually get your strain into culture. You then need to submit that culture to 2 different culture collections, of which there are many throughout the world. So ATCC is the American Type Culture Collection. DSMZ is out of Germany. KCTC is out of Korea. I believe JCM is out of Japan. So there are many of these. You just need to submit to 2 of them. I believe they need to be 2 culture collections from 2 different countries. You then need to write a peer-reviewed manuscript describing your new species and providing some information about that species. Most notably, what is the type strain of that species? Because as you've seen, this notion of type material is very integral to the rules of nomenclature. So at this point, your species name would be considered effectively published, but there is one more step here, and that is you need to notify the editors of the International Journal of Systematics and Evolutionary Microbiology. And they are basically the journal that is taking care of making sure that the rules of the code are followed. So you don't need to publish in that journal, but you do need to notify the editors of that journal that you have published this new name. They will then look at your publication, make sure it indeed does follow the rules of the code. And assuming that it does, they will then publish your name in what's called a validation list. And at that time, your name is validly published. All right, so let's now switch gears and talk about taxonomy. So again, taxonomy is concerned with how do we assign which organisms belong to a taxa? How do we draw these boxes around strains? And the most important thing to sort of realize here is that unlike nomenclature, there are no hard set of rules, and there is no sort of concept of an official taxonomy. Instead, what we have is taxonomic opinion, and that opinion is basically governed by best practices and convention, and the fact that if you want to express your taxonomic opinion, you're going to express that in a peer-reviewed manuscript. So you need to at least be able to convince, a set of reviewers that this taxonomic opinion follows the best practices in our field. So I'm sure all of you are aware of Meyer's biological species concept. So this is the idea that we can define a species as a population of organisms that can reproduce with one another, but that are reproductively isolated from other populations. So this is sort of a very widely used species concept for eukaryotes, but of course this doesn't apply to bacteria and archaea, which reproduce asexually. So what is the sort of equivalent definition for prokaryotes? And unfortunately, I think the short answer is that there's still no consensus within the community, and this is still very much an active area of research. So I just wanted to give you sort of some examples of different species concepts. So these are basically models that try to explain how do bacterial and archaeal species actually arise in nature. And is there some underlying mechanism that we can point to that says this is how species arise, so this is natural, so this is how we should actually define and use species in our taxonomy. So I'll just quickly go through these. So these are, this figure is from a manuscript by Ford Doolittle and Thane Papke. It's a great manuscript. If you're interested in this topic, I highly recommend you give it a read. So the first model here is the ecotype model. And what it's illustrating here is this black line is indicating that we have some populations, a group of cells, and they are maintaining genetic cohesion by the fact that every now and then there's a mutation that happens in this population. This mutation is very beneficial. So that cell is able to reproduce faster than everybody else in the population, to the point where there's a genetic sweep. Essentially, everybody else goes extinct, and we're just left with cells that have this new mutation. And because we have this periodic genetic sweeps, this population maintains genetic cohesion so that we always see it as a single species. Everything is similar enough there that there's no reason not to believe it's a single species. And then what the model proposes here is that some event occurs, some mutation, let's say, or just some stochastic process occurs, where some subset of this population ends up occupying a new niche. So that's what's indicated by these gray bars. And now, since we have these two populations and they're no longer interacting, they're going to pick up just mutations over time. And at some point, which is dictated by this blue bar here, they will have drifted enough genetically that we will then consider them to be different species. Okay, the second model here is a sort of similar idea. What's being depicted here is that, again, we have a population, but here we're imagining that we have this population and lots of recombination is happening. And because we have that recombination, this population maintains genetic cohesion. And then again, some event occurs, perhaps some mutation, where some subset of this population is no longer recombining with the other subset of the population. And then now that these populations are no longer recombining, they will pick up mutations and drift apart from one another to the point where at some point we will consider them to be separate species. So you can see that these are sort of similar ideas in that we need some mechanism that maintains genetic cohesion, and then some mechanism that allows there to be a barrier to gene flow so that populations can diverge to a point where we consider them to be separate species. Now, the last model here is the basically like a null model, and it proposes the idea that maybe there is no underlying biological mechanism giving rise to archaeal and bacterial species. And really what we observe today with our sort of extant taxa, or the strains that we see, is just the result of a stochastic process where some lineages go distinct for various regions and lineage diversify for various regions. So if you actually simulate a process like that, you actually can get trees that look very similar to the trees that we do observe. And of course, even though this process is stochastic, you do find places where you have a bunch of strains that are similar, and places where you have strains that are very distinct. And we can, of course, pick out places in this tree where we can say that we call this a species, but perhaps there just is no underlying biological mechanism. So that is certainly a possibility. Okay, so I just wanted to emphasize this distinction between sort of a species concept and a species definition. So I'm actually going to just read out this quote. This is directly from this paper by Ford Doolittle and Thane Papke. So what we want from a species definition is a set of easily applied and stable rules by which to decide when two organisms are similar enough in their genomic and or phenotypic properties to be given the same name. The needs for such a guide to taxonomic practice in medicine, biotechnology, and defense are obvious. And this is what I think the most important part is here. And even arbitrary rules to satisfy them would be better than no rules at all. We look to a species concept, on the other hand, for a genetic and or ecological model of bacterial diversification and adaptation. Ideally, this model would make sense for our definition, justifying the choice of one particular set of rules for defining species as less arbitrary or more natural than another. So just to sort of paraphrase that, the goal of a species concept, as we've just discussed, is basically to propose a model that describes why speciation actually occurs in nature, whereas a species definition is a much more practical thing. It's just, what are a set of rules that we can use to assign strains to species? And ideally, there would be some species concept underlying this, but just a set of arbitrary rules actually might be okay as long as those are applied in a consistent way and the species that result from those are useful to us for communicating diversity. Okay, so I'm just gonna go over one species definition because this is sort of by far the most widely used definition in our field, and that is the polyphasic species definition. So the idea here is basically that we should bring all the information we have to bear to decide what strains belong to a species. So in particular, we should look at genomic information, and the strains really should have high sequence similarity if they're going to belong to the same species. And importantly, this is something we can quantify. So we can measure this using DNA hybridization, 16S RNA similarity, or if we have full genomes, we can use something like average nucleotide identity. Now, we would also like strains from the same species to be monophyletic. Again, we can quantify this because we have ways now, of course, to infer the evolutionary history of organisms. And we would also like strains from the same species to share some sort of trait that can serve as diagnostic of that species. Now, this is maybe more of a sort of historical requirement that, it's not that long ago where having access to sequencing technology wasn't that common. So it was very useful if you had some easily identified phenotypical trait like morphology, or whether something was gram-negative or gram-positive, that could be used by clinicians and researchers to determine what strain a species belongs to when you have a bunch of sort of similar species. Now, one thing you will notice about this definition is it has no hard criteria and no formal rules of how you combine these different types of information. And again, this is basically left up to the community to decide. So this sort of gets quote unquote enforced by community consensus and the process of peer review, and you needing to justify to your peers that assigning these strains to a species makes sense. All right, so I just wanted to give you a bit of a historical perspective so you can see that the way that strains get assigned to species has really been driven by the molecular techniques we have at hand. Essentially, this comes down to making the best of the information that you have available to you. So in the early days before the 1960s, this was essentially getting done based on morphology, what media strains would grow on, and what was known about their pathogenic characteristics. And of course, with the benefit of hindsight, we know this is very limiting. There isn't that much variety in morphology, and it's very, of course, hard to test all the different conditions under which strains might grow. But this was sort of the information that people had at the time. So that's what basically taxonomy was being based on. Now this started to change in the 1960s, where we have DNA hybridization. So this is basically a laboratory method that allows you to take two strains and get an estimate of how similar those strains are. And in the 1970s, we start using 16S rRNA, and the big sort of advantage here is that we can start using that gene to infer the phylogenetic relationships between strains. And then in the 80s, this sort of matures to the point where we start using 16S as a criteria for delineating species, both by looking at sort of 16S similarity, it's really probably more to define higher order taxa, but also inferring the phylogenetic relationships between strains. And as you can imagine, this resulted in a lot of reclassification and proposal of new taxa, as we were able to determine that, how we defined these strains before doesn't actually line up with our modern view of these strains, where we can see their phylogenetic relationship. Now in the 2000s, sort of the big advent here is that we have cheaper sequencing technology and far wider access to more powerful computers. So this allows us to start getting full genome assemblies, and people start using concatenated protein trees to get sort of more resolved phylogenetic inferences. 2005, we have average nucleotide identity being proposed, and basically being proposed as a replacement to DNA hybridization, which, as I understand it, is a somewhat laborious thing to actually try and go do in the lab. It's obviously much easier just to get a computer to sort of calculate how similar your genomes are. And arguably, this sort of concept of average nucleotide identity starts to become favored over using 16S similarity when you actually have the genomes. So this is, of course, a big caveat that you don't have to go that far back, and you only have a very limited number of genomes, and that makes it hard to base taxonomy off of using genomes. But that starts to change in the 2010s, where we now have MAGs and much reduced sequencing costs. So we're getting lots of isolate genomes and all these MAGs from environmental samples, which pretty much brings us to today, where we have enough genomes that at least myself would argue that we can start building a taxonomy off of full genome sequences. And I think many in the field now agree that average nucleotide identity is sort of the gold standard for determining the similarity to different strains. And again, this is starting to result in a number of reclassifications as we start using ANI and these concatenated protein trees to infer the phylogenetic relationships between strains. Okay, so since we sort of have average nucleotide identity as being the gold standard, I wanted to just sort of briefly go over what that actual measure is. I think it's pretty straightforward to sort of understand at a high level what is being measured here. So what I'm showing you here is just two genomes, so one genome at the top here and one genome at the bottom. And then these regions in blue are regions that you can match between these two genomes. So that's what's sort of dictated by these arrows. And then there will be regions in these genomes that you can't match. So you can imagine we have some small region in red here where there is just no similar region in this other genome. And what average nucleotide identity is basically you take one of these matched genomic regions, you align them so that columns indicate homologous characters, and then you just count up the number of base pairs that are identical and the number of base pairs that are different. So in this case, you can see that 9 of the 10 are the same. So we'd call those regions 90% similar. And if you do that across all the blue regions, that is what average nucleotide identity is. Now there's a related concept called alignment fraction, which is simply the length of your genome. So that's all this section here. But it's all the matched regions, so taking all the blue regions here and dividing that by the total length of your genome. So it's just how much of your genome can you align to the other genome. Okay, so average nucleotide identity is basically a concept, and there are a number of different implementations of that. So originally, it was posed back in 2005. It was proposed using BLAST as the method to determine if two regions were similar, and criteria was put forth to decide, are they similar enough that we consider that a match or not? Since then, other methods have been proposed. So for example, there's ortho-ANI, which is also a BLAST-based method, but makes some refinements so that it runs much faster. There's ANI calculator, which I'm just pointing out here because this is a nice interface to a BLAST implementation you can get on a web server that's more comfortable for you to use. There are also methods that instead of using BLAST, they use alternative methods to identify regions of high sequence similarity. So there's MUMMR-based methods, which use a method called MUMMR. And more recently, there's been efforts to try and get faster methods for calculating ANI. Now that we have hundreds of thousands of genomes, calculating ANI can actually become a bottleneck. So a method that I often use is called FastAi that came out in 2018. Now what the little image is depicting here is just the correlation between a BLAST-based ANI on the x-axis here and FastAi is shown by these blue circles. And I just wanted to point out that these methods are highly correlated. This generally is the result for all these different methods of producing ANI that, of course, they're not identical, but they're highly correlated. None of these methods is more correct than the other. They're all using heuristics to find similar genomic regions. We tend to use BLAST as the gold standard simply because it was produced first, and we'd like to have some continuity in this measure over time. But all these methods, I think, are perfectly acceptable to use. I tend to use FastAi simply because it's fast. All right, so we sort of talked about the polyphasic species definition sort of in a general sense. And what's nice about this definition is that it is based on things that we can quantify: genomic similarity, evolutionary relatedness, phenotypic properties. But how do we actually apply this in practice? And it can be challenging because there aren't sort of strict criteria put forth on the importance of these different criteria or what exact values need to be achieved before you consider a strain belonging to a species. And arguably, this is both the sort of strength and the weakness of defining species in this way. So the strength is basically the criteria used to assign strains to species is left up to individual researchers who can then apply their expertise appropriately for the set of strains that they're studying, and they can organize those into species in a way that follows these guidelines, but also allows them to communicate their scientific results in the way that is best for them. So that can be a real benefit, that you have sort of species defined in a way that sort of highlights things that are of research interest. But this is also sort of the weakness of approach, that individual researchers are basically defining species as they see most suitable for what they're studying. And of course, what they're studying might not be of what's of primary interest to other researchers. So we have this sort of lack of consistency across the tree where how a species is defined in one part of the tree does not have to match how it's defined in another part of the tree. So it's sort of the double-edged sword here of leaving these opinions up to individual researchers. So I just wanted to sort of give you an example of what this might look like for defining species. So this is essentially the ideal case here, where we have a set of 10 strains. These strains are sort of laid out in a plane here. The idea here is that the distance between these strains is an indication of their genomic similarity, say measured using average nucleotide identity. And this is basically the ideal case. You can see that we have these clusters of strains where their intraspecific genomic similarity is very high, but the interspecific genomic similarity is relatively large. They're also distinguishable based on their morphology. Because they're sort of genomically so distinct, we can pretty much presume when we do a phylogenetic inference of these that these will come out as monophyletic. Now, a slightly more complicated sort of situation might be like this, where again, we have these nice clusters of strains and they're genomically quite distinct. Again, let's just assume that they come out as monophyletic, so they certainly satisfy those criteria, but perhaps there's no clear sort of phenotypic trait which allows us to easily decide which of these strains belong to which of these species. So you can imagine that, you know, all these have the same morphology, perhaps they're all gram-negative, they're all say methanogens, and undoubtedly if we can find some characteristic that is different between them, they are clearly genomically distinct, but ideally we would find some phenotypic trait that is useful to clinicians and researchers using the technology that they have on hand, right? So we want something that can easily be measured in a lab and applied for the practical purposes of deciding what species a strain belongs to. Now, as you might imagine, reality often looks more like this, where we have a bunch of strains, and these strains actually form a bit more of a genetic continuum, and it's actually not really clear how to cluster out these strains based on just their genetic similarity. You can imagine that this also presents a challenge because, of course, we have different ways to infer the phylogenetic relationships between strains. We can use 16S, we can use different sets of genes and concatenated protein trees. We have many models of evolution, and those may give us very different relationships in terms of the evolutionary relationship between these strains. So how to synthesize all that information can be a challenge. And over these set of strains, of course, they have many phenotypic properties, and we are sort of left with this question of, well, which of these phenotypic properties is the most important? How do we select between them? So this is sort of a practical problem. So one approach that has been proposed is, again, to go back to this concept of relying on type material. So this is actually something that was proposed in 2018 by Chung and his colleagues. And the idea here is that since we already have this system of nomenclature where we have the type strain of a species, and that sort of roots our nomenclature, perhaps we should also use that to root our taxonomy. So what you're seeing here is those same set of strains with just the type strain of species highlighted here. And then the idea is that you will just use a measure of genomic similarity. So again, perhaps average nucleotide identity and some criteria of how similar a strain needs to be to the type strain for it to be considered part of that same species. So this will basically allow us to pick out these 3 species, and then all the other strains which aren't similar enough to an existing type strain, those indicate strains that represent new species. So we'd have to define a new type strain in order to encompass those. Now, this is a very pragmatic approach to defining species, but it does have some limitations. And the major limitation here is that there is nothing really special about the type strain of a species. In fact, it's essentially a historical artifact. So you can imagine that, you know, at some point, instead of having all these strains, maybe we just had this little set of strains and a few strains here and a few strains here. And at that point, actually it was very clear from this point of view of strain diversity that we had 3 species, but the strain that was picked out as the type strain was more than likely simply the strain that was discovered first. So it needn't be the most typical strain or average strain or anything like that. So this again is sort of like, it's a pragmatic approach, but it's basically historically motivated much more than it is by any sort of underlying biological mechanism. And this sort of goes back to this, what was proposed by Doolittle and Papke, that, just having some rules is necessary basically to move forward. Now, I don't think these are arbitrary rules. Genomic similarity is a very powerful concept because we know things like phenotypic properties and evolutionary relatedness are going to be highly correlated with the genomic similarity of genomes. So we are sort of, while we're not directly trying to satisfy those definitions of the polyphasic species concept, we know we're going to get something that largely goes towards satisfying those criteria. Okay, so this sort of does leave the question of how do we decide what this criteria is for defining when a strain is close enough to the type strain to actually be considered part of the same species. I'm getting a few sort of notifications in the chat on my end. If one of the moderators just wanted to speak up and let me know if there's something I should be aware of, that would be much appreciated. Okay, so how do we answer this this question? And this is actually something that there is some level of consensus, I would say, in our community. And this sort of has a sort of a long standing in history. So this is basically going back to this DNA hybridization, which was introduced, I believe, back in the 60s, but there was a paper that came out by Wayne and colleagues in the 80s that basically proposed that 70% DDH be used as a criteria of 2 strains being similar enough that we should consider them to be in the same species. Now, again, there's no sort of like strong underlying biological mechanism to justify this 70% criteria. It is much more a case of this was a value that was found to satisfy species as they were currently being defined at the time. So it's sort of the value that just fit with the data that they had. And this criteria, because it sort of became a standard and became commonly used, was translated over to an ANI criteria in 2007. So that is what basically this plot is showing, that 70% DDH corresponds basically roughly to 95% ANI. Now, what you can see here is that this translation is, of course, not perfect. You do have cases where you have strains where their DDH is below 70%, but their ANI is actually above 95%. And basically just point that out to say that, again, these aren't really sort of magical criteria. They are somewhat arbitrarily selected, but that is potentially fine, right? As long as they allow us a practical way forward to define what strains belong to species, and we find that useful as a way to effectively communicate about diversity, they are basically serving the role it needs to serve. So this is sort of a different view of that data. So this is from the FastAi paper by Jane and colleagues in 2018. And here they're looking at, we have much more genomes at this point. So they're looking at 90,000 prokaryotic genomes and all the pairwise combinations of those, and plotting that out as a function of their average nucleotide identity. And what you can see here is that strains in the same species generally have an ANI above 95%. Strains in different species have an ANI below 95%, generally below, say, 84%. And then they sort of highlight the situation of E. coli and Shigella, which are, for historical reasons and clinical reasons, we maintain as separate species, but I think a lot of people agree from a genomic perspective are actually one species. Now, one interesting thing about this plot is you can see that there's a large discontinuity between about 95% and 84%, which does raise the question of whether or not there is some actual underlying biological mechanism that results in there not being sort of pairs of strains within this range, which might suggest that 95% ANI maybe does have some actual biological significance. I'm not entirely convinced of that myself, but it's certainly an open question and something that the community continues to explore. All right, so I wanted to give you an example of a sort of very atypical species, so you can see sort of how the application of the polyphyletic species concept can result in species being defined in very different ways. And again, I think this is both sort of the strength of the method and its weakness. So this is Brucella here. So within Brucella, there's actually 11 species, and these have been defined basically based on the host in which the strain was recovered from. So we have Brucella ovis, which is found in like sheep and goats, Brucella canis, which is found in wolves and dogs, and so forth. Now, if you look at this from a genomic perspective, actually, the vast majority of these strains are actually more than 99.5% similar in terms of average nucleotide identity. And the most diverse strain here is still 97.5% similar. So from a genomic perspective or an ANI-based species perspective, these would clearly be one strain. Now, perhaps from the point of view of a clinician, or it might be very sensible to define these strains based on the animals they affect. Now, I'm not an expert in Brucella, but given the high genomic similarity of here, it's really not clear that these strains are actually host specific. And sort of, I think, sort of illustrating that is that there are 4 different species that actually infect humans, as listed here. Nonetheless, this is sort of the current taxonomic opinion in the literature. And you can see, so this is basically just a valid taxonomic opinion, but it's one that is sort of extremely atypical compared to how we define bacterial species in other parts of the tree. All right, so I just wanted to briefly touch on how we can assign strains or organisms to higher taxa. The issues here are largely the same, but I think there's a little bit less consensus here, which is why I really sort of focused in on species. So for delineating genera, it has been proposed that we can use the percentage of conserved proteins. It's also been proposed that we could use some sort of ANI criteria, so sort of like 95%, some lower bound like 85% or something like that. What I would say is that I think a lot of genera are still sort of defined historically based on their phenotypic characteristics, which is what people could sort of measure and observe at the time. So for example, Bacillus is largely defined to be gram-positive rod-shaped organisms. Because of that, there have been lots of strains and species defined within Bacillus. Now at higher ranks, it's been proposed to use average amino acid identity. So that's sort of similar to average nucleotide identity, but looking at the amino acids on proteins instead. And 16S thresholds have been proposed. Now these have been proposed sort of fairly recently, so they, of course, just haven't had a chance to be applied to a lot of hierarchical taxa. Now, of course, a lot of the hierarchical taxa we know were proposed sort of in the past before these criteria were put forth, and again, are basically based on sort of key shared metabolic properties. So we have things like Methanobacteriaceae, which are a group of anaerobic organisms that perform methanogenesis, or cyanobacteria, which is a phylum of organisms that perform photosynthesis. Now, just like species, these higher taxa are, of course, a matter of taxonomic opinion about how you want to decide which organisms actually belong to a taxa. So I just wanted to give you a sort of concrete example of that. So what you're seeing on the right here in this tree is we have a set of lineages here, and these are all classes of cyanobacteria. And recently, in the 2010s, we've discovered this whole new branch here of organisms, which have been called melanobacteria, which sit sister to these cyanobacterial classes. So the question here is, and sorry, the root of the tree is up here. So the question here is, so these melanobacterial organisms, should we consider them the most basal class of cyanobacteria, or should we consider them to be a new phylum? Now, what makes this question particularly interesting is that the photosynthetic machinery is actually acquired here. So these organisms are all photosynthetic, whereas the Melanobacteria are not. Now, both opinions have been put forth in the literature, and I just wanted to go over that so you can see that indeed, there are differences in taxonomic opinion, and this is sort of part of the process in our field. So the first paper here is basically this paper by Su et al. From 2014. Full disclosure, I'm a co-author on that paper. And this paper takes the point of view of adopting this definition by Phil Hugenholtz, which was proposed back in 1998. And the idea here is that a new phyla should be a monophyletic group that is unaffiliated with existing phyla. And the rationale being we want to keep the number of proposed phyla to a minimum, and a phyla should really mean a sort of a line of primary descent that doesn't consistently associate with some other group of organisms, because if they do, then we should combine those all into a single phyla. And indeed, that is the case here. So if you infer a bunch of different trees between these organisms and look at their support values, you find that Melanobacteria consistently associates with these cyanobacterial classes. So under that sort of rationale, in this paper, it's proposed that Melanobacteria be considered the most basal class of the cyanobacteria. Now, the opposite conclusion has been proposed by Derenzny et al. I'm sure I'm butchering that name, my apologies. And this argument is basically based on using 16S as a criteria for delineating phyla. So they basically looked at the 16S sequences in the cyanobacteria classes, compared them to the 16X sequences in the Melanobacterial organisms, observed that most of those were below 85% sequence similarity, and on that criteria argued that Melanobacteria should be considered its own phylum. So both of those are, of course, perfectly valid taxonomic opinions. They both put forth rational arguments. I think there's also a very valid argument for just keeping Melanobacteria as a separate phylum, purely for the fact that we can use cyanobacteria to mean a group of organisms that are photosynthetic. So all those are valid taxonomic opinions, and this is basically encouraged in our community, but as you can imagine, also does lead to some level of confusion where there's not, of course, perfect agreement, and we need to be able to deal with that sort of level of uncertainty. All right, so that pretty much wraps up what I wanted to talk about in terms of an introduction here. I just wanted to sort of leave this section with this slide, just so there's some slide indicating all the different resources you could go consult. So in terms of nomenclatural resources, we talked about LPSN. I think that's sort of the most common resource and where I usually go first. There's also Berge's manual. So this used to be a large book that people will actually browse through. These days, of course, it's a website. And there's Names for Life, which also has basically much of the same information at LPSN. It can be good to consult multiple of these resources if there are cases where there may be reason to believe there's uncertainty in the information being presented. Of course, these resources are all maintained through the hard work of people in our community, and mistakes do occasionally get made. There are also a number of taxonomic resources, so we're going to talk about the Genome Taxonomy database in some detail here. At this point, I'll just say it's a genome-based taxonomy, and it covers taxonomic classifications for bacterial and archaeal genomes. So it sort of has a very specific scope. There's the NCBI taxonomy. This covers, of course, all domains of life and is applied not just to full genomes, but also 16S sequences and basically all the different genetic information that they have in their different repositories. If you're interested in 16S or 28(3)S sequences, the most common resource these days is Silva. And then if your research takes you into other domains, there's UniUke, which is sort of an initiative somewhat similar to GTDB in that it's sort of using a reference tree to redefine eukaryotic taxonomy. I believe that initiative is currently focused on protists. And if you're interested in viruses, there's the ICTV. Now there also is a number of tools for doing taxonomic classification. These are all tools that are specifically aimed at classifying genomes. And basically I'll go over these sort of briefly at the end of this talk and just sort of contrast the different information that they provide you. All right, so this sort of, I'm going to sort of switch gears a bit here and sort of get into the GTDB and basically what does taxonomy and nomenclature look like today, and try to get into try to make it clear to you why there isn't a sort of complete agreement in our community in terms of the best way forward. And again, this basically stems out of the fact that we are getting a flood of new megs, and these megs represent a lot of uncultivated diversity, and we would like to organize that diversity. So this is, for example, is a paper published in 2018 by Tolley and his colleagues, where they recovered about 2,500 megs. It's a paper by myself back in 2017, where we recovered about 8,000 megs. And then there was a series of papers in 2019, all looking at the human microbiome, where they're literally pulling out tens of thousands of megs and showing that these actually represent tens of 10 or 20,000 sort of new species that sort of are yet to be described in a formal sense by science. Now, of course, on the whole, this is great news, right? This is basically giving us access to the uncultivated microbial majority at the genome level. So we've sort of known that there's all this hidden diversity for a long time by looking at 16S sequences, but now we actually have their genomes, which provide us, of course, with much more information about what these organisms are doing. However, I think it is safe to say that we weren't really prepared for this flood of new genomes and how to best handle them. And basically this comes down to, again, this fact that like, this is great, we have all these new genomes now. In fact, we have so many genomes now, it starts to make sense to actually characterize this diversity in a formal taxonomic and nomenclatural sense. But as I'll argue here, basically that perhaps these sort of systems aren't up to keeping up with the pace of new discovery. So I'll just take you back to this slide about naming a species, and particularly just the first part here, that if you want to name a new species, the first thing you need to do is get a strain into culture. And of course, this is basically a non-starter if the species that you know about comes from a mag. Now, of course, you can try and get that that species or strain into culture, and people are, of course, doing that and developing new techniques to do that. But the simple reality is that takes a lot of effort. It often isn't successful, and it requires a sufficient amount of effort that it's basically not keeping pace with the rate at which we're discovering new species. So this, of course, was recognized in the community that this process of needing to do cultivation is basically a bottleneck in terms of being able to formally put forth new validated names to describe species. So this is basically what sort of gives rise to the seek code. So I'll just sort of take you sort of briefly through the history of this so you can appreciate where it comes from. So basically the story starts back in about 2005, where Barney Whitman sort of recognized this bottleneck and proposed that, well, one way around this is that we could modify the current code so that we could use DNA sequences as type material instead of requiring it to be something that's in culture. This basically spurred a lot of community discussion. So I'm leaving out basically 5 years of community discussion here. There were a bunch of papers proposed, there were workshops held, and the ultimate result of that was in 2020, this consensus statement was put forward. And in this statement, basically it again argues that, yes, this is a huge bottleneck in terms of trying to name this uncultivated majority. We would like to be able to formally name this and apply formal taxonomy to it, and that there are an appreciable number of people in our community that are of this opinion. So this is basically the main purpose of this consensus statement by Alison Murray, is that it has 63 co-authors and 50 other people in the community that sort of endorsed this idea of having DNA be suitable type material. Now, unfortunately, in 2020, the ICSP, so this is the committee that governs the prokaryotic code, rejected the proposal to accept DNA as type material. Now, there is very good arguments for doing that. The main one, I believe, being put forth is that we would actually like to have type material in culture so that biologists can go to their culture collections and order these and get them in their lab and do actual biological experiments on them. So, there is certainly a rationale for not using DNA as type material. However, there is sort of this segment of our community that still believe, well, that's fine, and we should still try to get things in culture, but there is a need to have a way forward, to have a formal nomenclature for the uncultivated majority, where a lot of these things may not get into culture in the foreseeable future. So this has sort of given rise to an effort to put forth a new rule of nomenclature, which will be called, well, it's being called the SEEK code right now. And this is basically being actively worked on in our community, and I expect will sort of come out in a more formal sense sometime this year. Now, sort of related to this idea of dealing with this flood of new diversity, we can also sort of look at the NCBI taxonomy and the GTB taxonomy. So the sort of mandate of the NCBI taxonomy is to reflect the latest taxonomic opinion of our community as is expressed in the peer-reviewed and published literature. And the GTB sort of takes a different view on that, where basically here we're trying to aim to, of course, reflect the latest taxonomic opinions, but we're also trying to build a more quantitative framework. So we have quantitative criteria that taxa need to be rank normalized, which I'll explain later on in this lecture, and that we should actually infer a tree and make sure that taxa are monophyletic or phylogenetically consistent on this tree. Now, the GTB also differs from NCBI in that it's strictly genome-based, the rationale here being that we now have a sufficient number of genomes, and that genomes are the best genetic description we're ever going to get of a strain, that we should embrace that and start building our taxonomy off of that information. And sort of the underlying rationale here that because we are discovering new diversity so rapidly, we need to come up with a taxonomic framework that's more formalized, in the sense that it can be literally codified, in the sense that we can write code to help us actually define new taxa at a rate that is practical, in the sense that it doesn't require too much manual effort and isn't sort of bogged down by the fact that every time you want to express any sort of change in taxonomic opinion, you need to write a full manuscript. As I'm sure everybody here is aware, publishing a manuscript is a very time-consuming process. So that's sort of the background rationale for the GTB. So now I'll sort of take you through, sort of practically speaking, what is the GTB doing, so you can get a sense of what it is, and also how it applies these rules of sort of nomenclature and taxonomy, well, ideas of taxonomy. So very similar to what I presented before, species in the GTB are defined by considering the average nucleotide identity to type strains. So this little flow chart illustrates sort of in a high level what is done. So we are taking genomes from the NCBI assembly database. We filter out poor quality genomes. These are generally genomes that we don't believe we can robustly put into a phylogenetic tree. We then identify which genomes are assembled from type material. So we do that by consulting LPSN. There are then some subset of genomes that have recognized species names, but where the type strain of the species hasn't been sequenced yet. So we have some sort of set of rules that allow us to select sort of a representative for those species, which effectively acts as the type strain until the actual type strain is sequenced. We then form species clusters around these named species, and then we're left with all the genomes that don't belong to any existing named species. So we organize those into de novo species clusters and give them placeholder names, so that we have some way to communicate about them and hopefully can find which of those species are important. Are there sort of species in there that are correlated with diseases that we really should study more and eventually actually give proper Latin names to. So I'll just walk you through how this process works in the GGB using this sort of conceptual example. So what I'm showing you here is each of these gray squares is a genome. The distance between these squares is an indication of their average nucleotide identity. So again, how we start here is we identify all those genomes that were assembled from the type strain of the species. This can be done by consulting LPSN and matching up those co-specific strain IDs. So in this simple example, I'm just showing you 6 type strains. Now the typical example and most forward to understand is these type strains that are well separated from one another, where we just apply this 95% ANI criteria that we discussed. So any genome that has an ANI greater than 95% is assigned to that type strain and is classified as being from the same species as that type strain. So this is the vast majority of cases. However, we also do have cases like this, where you have 2 type strains that are more than 95% similar to one another. And in this case, because they're 96% similar, so similar but not too similar, as I'll discuss in a second, we actually keep those as separate species because we'd like to recapitulate the existing taxonomic opinion of the community to sort of the extent possible, while still trying to sort of have some normalized or standard definition of what a species is in the GTB. So you can see this is sort of very few cases, sort of less than 400. And sort of the rule here is strains will now be assigned to these type species, will be assigned to the closest type strain, but the ANI radius here actually gets reduced to reflect the difference, the distance, the ANI distance between those 2 type strains. And the rationale there is that just helps ensure that those strains are likely going to be monophyletic when you infer the phylogenetic relationship between these. Now, the last case here is when you have 2 type strains that are actually highly similar to one another, which in GGB we define as greater than 97% ANI. In this case, basically, at this point, we basically decide, no, these things are too similar. We no longer think they should actually represent separate species. So we basically use the rules of nomenclature to determine which of these species has priority. We retain that type strain, and the other species becomes a synonym of that retained species. Now, so this sort of deals with all the strains that are similar enough to a named species to be assigned to one of those species. But as you can see, of course, we're going to be left over with some genomes. And these are preferentially going to be genomes that represent sort of this uncultivated majority. And so basically a bunch of MAGs. And the way we deal with these is we do what's called greedy clustering. So we order these genomes from basically highest to lowest genome quality. I'm not going to go over the exact formula of how we calculate genome quality, but it's just a combination of sort of the measures you might expect, an estimation of their completeness and contamination, how many contigs are in the assembly, and sort of maybe importantly, we do preferentially select isolate genomes over MAGs or SEGs. So given this sort of measure of quality, we can figure out which of these unclustered genomes has the highest quality. So let's say it's this one. It becomes our representative of a new GTDB species, and then we just apply the standard ANI rules. Then we find the next genome that has the highest quality amongst those that are still not in a species cluster, and we continue doing this process until all genomes have been assigned to a species cluster. So one thing we can look back on now is what happens to Brucella. And as you might imagine, under this sort of more strict genomic definition of what a species is, these 11 sort of traditionally defined Brucella species become a single species in the GTB. It's called Bacillus melitensis because that is the species name with priority, and the other 10 species are synonyms of this species name under the GTDB framework, right? So this is, of course, a matter of taxonomic opinion. So this is sort of a higher look at these GTB species clusters. So on the outer ring of this pie chart, it's showing for these GTB representatives, is the representative something that was actually assembled from the type strain? Is it something that has a Latin name, but where the type strain hasn't been sequenced yet? So we had to select a representative. And what you can see is actually the vast majority of, GTB species clusters actually have placeholder names, and placeholder names look sort of like this, where they belong perhaps to a recognized genus, but it's a new species, or perhaps it's even a new genus. So these also just have alphabetical alphanumeric names. Now the inner ring here is showing whether or not that representative genome is isolate, which you can see it is in the vast majority of the cases, or whether it's a MAG or a SEG. So you will notice that there is actually a bunch of cases where we have placeholder names and the actual representative genome is an isolate. And this largely occurs because there are cases where using this ANI based definition, you have species that say span everywhere down to 90% ANI. So they're all considered part of a certain species. And we've actually broken that up into multiple species. Okay, so one thing to sort of appreciate here is that using this scheme, we're able to give all genomes a complete taxonomic assignment from species all the way up to domain. Now, a lot of those names are placeholder names. They'll still allow us as scientists to communicate about that diversity. But if those species or any of those taxa become important, ideally, a name would formally be put forward. And you can contrast this sort of with the NCBI, where you have, so I'll just point out the example at the bottom here, where there are sort of genomes. In this case, it's a MAG, where it's actually only classified as a candidatus phyla, and there's no more specific classification to this. So this is sort of one sort of aspect that the GTB tries to address, is that we would really like to divide all the genomic diversity that we know we have up into species clusters so that we can talk about it in a much more resolved way than we currently can, because right now, because of limitations in the prokaryotic code and the pace at which new taxonomic opinion comes out, a lot of diversity just hasn't been described to that level of resolution. Okay, so that's, we've been talking about how we basically assign genomes to species clusters. So in the GTB, what do we do for higher ranks? And basically, the GTB has two objectives above the rank of species. So the first is that we should resolve any polyphyly that exists, and that we should normalize taxa that are at the same taxonomic rank. And we basically address the first of these by inferring a concatenated protein tree and ensuring that taxa are actually monophyletic on that reference tree. And we normalize taxa at the same taxonomic rank using a measure called relative evolutionary divergence. So I'll go through sort of each of these objectives and how we actually achieve them in practice. Okay, so this we're going to start with how we resolve polyphyly. So what I'm showing you here is the GTDB reference tree. So this is the bacterial reference tree. It's inferred from 120 concatenated proteins. And what you can see here is basically two very conspicuous genera. So these are basically genera that it's sort of been known for a long time that they are not monophyletic. And indeed, that's exactly what we see on our reference tree. So I'll just focus on Clostridium. So what you can see here is that we have a bunch of different Clostridium lineages shown in this orange color. So how do we handle this in the GTDB? So first of all, we have to go back to this concept of type material. We need to identify which of these lineages contains the type species for the genus Clostridium. This happens to be this lineage at the bottom. And then for all these other lineages, which are comprised of genomes which have historically been classified as Clostridium, we just append an alphabetical suffix. So we get Clostridium W, Clostridium Y, Clostridium V, and so forth. And there are actually 41 different Clostridium genera that are defined this way. So, far more than what I'm showing you in this subset of the reference tree. Now, ideally, like we're just giving these placeholder names with the idea that the community will independently verify these results and formally put forth actual Latin names that get validated under the prokaryotic code. And sorry, I should just go back here and say, this was basically the view of the GTDB back in 2018. And this is basically the view in 2020, where I just wanted to show that this process is taking place. So I guess there's 4 examples here of polyphyletic eubacterium or Clostridium lineages, where there have been independent research done, which has sort of agreed with the GDB that, yep, these do represent polyphyletic lineages, and these authors have put forth actual formal Latin genus names for these lineages. Okay, so now I'm going to discuss how we sort of do rank normalization and sort of what rank normalization is. So the idea here is that, right now, without any sort of formal and widely adopted rule for defining higher taxa, you have the same problem that we saw with species, that in different parts of the tree, what something, say, like a family means can mean very different things. So what we'd like to do is sort of normalize those in some sense, so that when we talk about a family, we know we're sort of more comparing apples to apples. And in particular, what we think is sort of a natural sort of definition for defining those is that taxa at the same taxonomic rank, so let's say all families, should have coexisted at the same time in the past. So I think it's probably easiest to understand that sort of just by going through these trees here. So on the sort of left here, this is just a phylogenetic tree where the branch lengths here indicate expected substitutions per base pair. And what we'd basically like to be able to do is take this tree and scale the branches, so that instead of expected substitutions per base pair, they actually measured years of divergence, right? So an actual measure of how far in the past did this internal node exist. So let's, if we imagine we can do that transformation, then we get an ultrametric tree where every internal node now actually has a date of when it existed in the past. And then it's very natural to say, well, all family should have coexisted in the past. And that's what's shown by these different rings. So the first ring here is saying this is the interval in which we believe all species should be defined. This is the interval in which we believe all genera should be defined, and so forth, such that those taxa will have coexisted. Now, unfortunately, in practice, it's sort of not practical to take a phylogenetic tree and scale the branches so they represent actual time of divergence. Basically, computationally, that's a very challenging problem. And on top of that, basically, we just don't have enough hard dates to actually even solve that problem. So, of course, this does get done for eukaryotes, but there, of course, we have a very rich fossil record. For bacteria and archaea, there's very few internal nodes where we can actually put a date on, which basically makes this problem sort of unresolved at the moment. So what we do is basically use an approximation to actual time of divergence, which we call relative evolutionary divergence. And that's what's illustrated here. So this is pretty straightforward. So basically we set the root at 0. So this is at sort of time 0. And then we take all the extent taxa and we set those to be at time 1. And then we linearly interpolate the relative evolutionary divergence of internal nodes such that they're basically taking into account varying rates of evolution, right? So you're taking into account the fact that this branch is actually longer than the branch over here, and taking that into account since the rates of evolution are different between these two lineages. Now, doing this linear interpolation is a straightforward formula. So I've given you sort of just one example here. If you're interested in the details, you can sort of look that up in the manuscript, but I just wanted to point out the simple fact that this is nothing more than addition and subtraction, multiplication and division, right? So there's nothing super complicated going on here. And what this does allow us to do, though, is to basically take a tree, get this measure of relative evolution divergence, which we believe is a good approximation of actual time of divergence, and then use that as a way to normalize taxa so that they will have roughly diverged at the same time in the past. So how do we actually quantify all that? Again, we want sort of very practical rules here that we can actually quantify and use as a guide for defining taxa. So what we have done is basically we've looked at all taxa ranks, say all families, and determined the median RED of all those families, and then defined an interval of plus or minus 0.1 RED units around that median value. And there's a few reasons for that. So plus or minus 0.1 is a somewhat arbitrary chosen criteria, essentially chosen to be fairly wide for basically the three reasons I have listed here. So the first is that, as I've explained, RED, relative evolution divergence, is approximation for what we'd really like to do, which is have taxa coexisted at the same time in the past. But that's just not practically possible, so we're using this approximation. The second is that we would like to put taxa on stable internal nodes. So you need to put a family on an actual node in this reference tree. There are, of course, a finite number of nodes. Some of those nodes actually aren't that stable. They have low bootstrap support value. So we want that interval to be wide enough that more often than not, we do have a node where we can define that family or genus somewhere where we believe those organisms are going to be evolutionarily related under different phylogenetic inferences. And lastly, of course, we want to recapitulate existing taxonomic opinion to the extent possible, while still wanting to achieve this goal of normalizing sort of taxa that are major outliers, so that we can say, yeah, when we meet a family, we do mean things that are roughly existed at the same time in the past, and in that sense, we're sort of comparing apples to apples. All right, so this is sort of a view of NCBI taxa as it relates to relative evolution divergence. So what you're seeing here is relative evolution divergence along the x-axis here. So again, we have the root of the tree at 0 and extent taxa at 1. And then each of these little circles is an NCBI taxa at different phylogenetic ranks, or different taxonomic ranks. Now the blue bar here is the median value of those NCBI taxa, and the black bars here are these plus or minus 0.1 intervals. So again, we want to be able to retain as many of these names as possible. So when you view it this way, you can see why we're making this a fairly wide margin for including taxa. But you can see that there are still lots of outliers. And the goal of the GTB is basically to resolve these outliers. So there are basically two ways we can do that. So I'll just do that by example here. So here we have Nitrosporiceae. So you can see that this is way too close to the root to be considered a family. And there are basically two ways we can resolve this. So one, we could take Nitrosporiceae and we could break it up into multiple families. So those would all be closer to the leaves in our tree. So we can break them up to the point where there are multiple families that exist in the range that is acceptable in the GTB. Or we can recognize that Nitrosporiceae actually has an RAD value much more commensurate with a phylum. So we could reclassify this family as a phylum. So in this case, this is what we did. We reclassified this family as a phylum and these genera as classes. So you can see that you get this. You can also see this is sort of an illustration to the extent of which these things have been reclassified, right? So this is what we originally started with defining the GTB. And after we go through this process of normalizing all the taxa, we end up with something that looks like this. So you can see that what a family now means is a much more a consistent definition. Now, I will just point out that there are still some outliers. And again, this is just a pragmatic issue that there are some, very few number of cases where you have a class and a family and you have internal nodes that you put those on that satisfy your rule. But once you've done that, there just actually is no stable internal node in which to put that order on. So that just is sort of a pragmatic problem that happens in a few cases. All right, so the other way to resolve these RAD outliers is, as I described, you could take clostridales in this case and break it into many orders, or sorry, break it up into many different orders. Now, which of these two approaches we take is pretty much left up to sort of the expert opinion of the GDB curators, but often sort of the data in the tree dictates sort of the most obvious path forward. So in particular, clostridales is polyphyletic in the tree. So in order to also achieve the goal of things being monophyletic, it's just very natural actually to define each of those polyphyletic lineages as new orders. And that's sort of just depicted here, where we have the NCBI order, clostridales, and it has been divided into all these different orders within the GTB taxonomy. All right, so just to summarize the GTB here, it satisfies these three properties. It's complete in the sense that all genomes have assignments from species to domain. It's rank normalized. Species are normalized based on a definition of A and I. So all species, the strains assigned them are generally within 95%. In a few cases, that can be down to 97%. And higher taxa are delineated using this concept of relative evolutionary divergence. And then it's phylogenetically consistent in the sense that all taxa are monophyletic in the concatenated protein tree we use to guide the classification. So this is a comparison of the GTB and NCBI classifications over the about 32,000 species representatives in the GTB. So the blue bars here are indicating cases where we agree on the classification of that genome. So you can see that's less than 50% in the majority of different ranks here. The orange bars here are cases where there's been a passive change. So this is just somewhere where the GTB has a taxonomic opinion, but the NCBI just doesn't have a taxonomic opinion. So we're just sort of filling in information there. And then the green ones here are where there's an active change. So both GTB and NCBI are expressing a taxonomic opinion, but our opinions are actually different. Now, I think this plot actually makes it look like there's a ton of disagreement between the GTB and NCBI. But a lot of that disagreement is sort of cases like Clostridium that I showed you, where in NCBI, you have a bunch of lineages that are all called Clostridium, and they get broken up into 43 different lineages in the NCBI. So those sort of cases like that account for a lot of this disagreement. And sort of as a real sort of example of that, the reason there's so much agreement at the phylum level actually comes down to just changing the suffix of some well-recognized phyla. So in particular, the GTB, basically we are sort of probably adopted too early, actually the 2020 acceptance of putting phyla into the nomenclatural code. So since we follow that, we have redefined a lot of phyla to end with the suffix -oda. So in GTB, you have Aerophobota, but in NCBI, you have Aerophobetes. So these sort of minor changes to, follow the code or not account for a lot of these active changes. So we actually, I think, we're in more agreement than perhaps this bar chart would initially suggest. Okay, so now I just wanted to sort of switch over to sort of very practical discussion about the GTB website and GTB TK. So this is the homepage of GTBB. It gives you some just initial information, the number of genomes that it spans and how those genomes are broken up into different taxa. Really, the most important information on this page is the version number. So we're currently on release 5 of the GTB, And this release is based on genomes that were at NCBI as of when RefSeq 95 was released. So this is something you can look up at NCBI that has an actual date. So this version of the GTB encompasses all genomes that were at NCBI as of July 8th, 2019. Now we have a few different views of the GTB taxonomy. We have the standard hierarchical tree view, where we point out just some specific information relating to type material, whether or not something is the type strain of a species, the type species of a genus, whether or not a genome is the genome we selected to represent that species. And there isn't a case here, but we also use the hash symbol to indicate Latin names that we've proposed, but which actually aren't validated yet. This is something we sort of did in one of the initial releases of the GTB. These days, we actually no longer put forth Latin names, and we always use sort of alphabetical suffixes like this and leave it to the community to propose and validate new Latin names. This is perhaps the most useful view of the taxonomy for at least understanding how it's changed over time and how it relates to the NCBI taxonomy. So this is the taxon history view, and what I'm showing you here is the history for the family Eubacteriaceae. So on the right here, you can see how genomes that have been classified as Eubacteriaceae, either at NCBI or in any of the GTB releases, how they're named at NCBI. So you can see that the vast majority of those are classified as Eubacteriaceae, but there's one genome that just doesn't have a classification. So this is sort of somewhere where we've filled in a classification, and one genome here where, at least according to GTB, we would consider this genome to be misclassified. But what you can also see here is that Eubacteriaceae has been split into multiple different families within the GTB, and this is in order to achieve this goal of rank normalization. And you can also see that between release 89 and 95, the classification of genomes has been very stable in the GTB. We also have sort of a release statistics page. So this has a bunch of different visualizations and tables covering stuff like how many genomes does it cover, how many of those are bacterial, how many are archaeal, how many representative genomes are megs or isolates, things like that. The view I'm showing you here is just indicating the quality of the genomes that are selected as GTB representatives. So you can see that the majority of these actually are high quality genomes, but we do sort of aim to be inclusive. So we are using megs that are as low as 50% complete in order to try and cover as much genomic diversity as possible, while still only using sort of megs that are at least good enough that we know that they're going to give accurate ANI estimates and have enough marker genes that we can put them into phylogenetic trees. If you're a bioinformatician, probably the most useful section of the website is actually just these raw data files. So I won't go through all of these, but there is a file descriptions file which explains what all these different files are, and there are a bunch of other files in these subdirectories. There is a methods file which explains how this exact release of the GTB was put together. We do update version numbers of reference databases and versions of software more often than we put out formally published manuscripts, so you can consult that. And then we basically divide the main data files into archaea and bacteria. So in particular, you have the reference tree here that we use to define the taxonomy. We provide over 100 metadata fields for each of the different genomes that are in the GTB. And then there's just a flat file that actually has the GTB taxonomy string for each genome. Now we do have a forum for GTB, so we do recommend that if you have any questions about GTB or GTBTK, that you post it on this forum so that everybody in the community can see these kind of questions and benefit from the responses, and that other people in the community can then chime in if they have further questions or further insights into questions that are being asked. Okay, so that's sort of the GTB website. So I just wanted to end this talk by going over the GTB TK. So this is just showing where it fits into the sort of standard workflow that was introduced by Dylan. I think that was about 2 weeks ago. Where basically you start with raw sequencing reads, you QC these reads, you're going to assemble them and then bin them into mags, and then probably apply some sort of QC to those mags to find the subset that you think is good enough to do your study on. And this is where sort of GTB-TK fits in, is it answers basically the question, what are my genomes? You want to get some taxonomic classification for those genomes, and that is precisely what GTB-TK does. And then you can, of course, go on and do your genome results study. So GTB-TK is available on GitHub at the URL here, and there is full documentation there, but I will sort of just step you through the very basics of it here. So there are basically 2 workflows for GTB-TK. There's the classify workflow, and there's the de novo workflow. So the de novo workflow basically will identify marker genes in your genomes, add those to all the representative genomes in the GTB, and then infer a de novo fast tree. This is very computationally expensive and is, not the most common way to use GTB-TK. What's more common is what you want to do is use the classify workflow, where basically you have a bunch of genomes and you just want to get taxonomic assignments for those relative to the, within the GTB framework. And this is very straightforward. So you can just basically provide a directory that contains all your genomes. Alternatively, you can do, I think it's -- genome file or maybe genome batch file, and submit a very simple description of like where all your genomes are on your computer. GTB-TK then will take those and output the results in the directory that you specify. Now, one caveat here is GTB-TK is fairly resource intensive, so it does require about 150 gigabytes of RAM and takes about an hour to classify 1,000 genomes with 64 CPUs. Now, this doesn't scale linearly, so even if you want to do a single genome, you still need about 150 gigabytes, and I think it still takes something like 20 minutes. There basically just are some steps here that require a lot of memory and time, regardless of the number of genomes. We are working on alternative methods sort of to address that. In the meantime, if you don't have access to a computer with those sort of resources, GTB-TK is on K-base. So this is like an online web service that has a bunch of different bioinformatics tools that you can apply to your genomic data. So this is certainly worth looking at if you don't have access to more powerful computers, or if you're just more comfortable working on your data through a web interface. Okay, so this is sort of the command line output of the GTB-TK, and it basically follows the actual sort of workflow of GTB-TK. So I'm just going to quickly walk through this so you can get a sense of what it's doing. So essentially, it takes your genomes and identifies the genes within your genomes using a program called Prodigal. It then uses a piece of software called Hammer and HMMs for the TigerFam and PFAM protein families to identify the set of either 120 bacterial or 122 archaeal marker genes, which we use to infer those GTB reference trees. Once those genes or subsets of genes have been identified, those are concatenated in exactly the same order as what we do when we actually do the reference tree. And then we apply a mask to that concatenated alignment to cut it down to about 5,000 base pairs. And this is just to sort of keep computational requirements to some level of sensibility. Now you basically have multiple sequence alignment that's in the exact sort of multiple sequence alignment space as used for the GTB reference tree. So you can use a program called pplacer, which will determine what is the maximum likelihood placement of your genome in this already established reference tree. And that's what we then use to calculate the RED of your genome, the relative evolutionary divergence. And if your genome falls within a named GTB genus, we also calculate the ANI using FastAi to all the different species representatives to see if it satisfies the ANI criteria of actually belonging to an already defined GTB species. So there are a number of output files produced by GTBTK, including like a NUIT tree that shows where your genomes were placed in the reference tree. But for most people, the only output you need to consult is the classification file, which basically just indicates for your genome, what is the GTB classification of it. And the rest of these fields just indicate additional information that sort of explain why GTBTK came up with this particular classification. All right, so this is basically the last slide here. I just wanted to give you an example of classifying a single genome using a different classification tools. So these are all tools that I've used and can recommend. And I wanted just to sort of highlight the difference between them. So the test genome here is a genome that's classified as Bacteroideales at NCBI. So, something that's pretty novel. Indeed, this is a MAG, but it's a very high quality MAG. It has all of the sort of ribosomal genes. It has 20 tRNAs, and it's estimated to be less than 1% contaminated. So if you submit this genome to GTBTK, basically what you're asking to do is to determine the taxonomic classification of this genome, where species are delineated using ANI and higher ranks are delineated using relative evolutionary divergence. And the classification you get back is going to be relative to the GTB taxonomic framework. And in this case, you get back this classification. You'll notice that we agree with NCBI on the order of this genome. You can also use MIGA. So this is the Microbial Genome Atlas. So when you submit a genome to MIGA, you're basically delineating taxa using a combination of amino acid identity and ANI. So similar to GTBTK at the species level, they're using average nucleotide identity, but for higher taxa, they're using average amino acid identity. So you can see this sort of difference in how we can, this is sort of a difference of opinion in how we can circumscribe genomes to higher taxa. Now, importantly, MIGA, you'll get a classification that's relative to the NCBI taxonomic framework. In this case, basically, it will say with confidence that this genome is definitely within the class Bacteroidea, and that it's likely within this order Bacteroideales. And because this is relative to the NCBI taxonomy, it doesn't give you a more specific classification beyond that, because there is no more specific classification within the sort of existing NCBI taxonomy. You can also submit your genome to the type strain genome server. So again, this has a different methodology for how to circumscribe taxa. They use something called the genome blast distance phylogeny. Here, the classification, again, is based on the NCBI taxonomy. Now, this service is really geared towards type strains. So in this case, because this genome is very novel, basically what it tells you is the most similar type strain is this Bacteroides species here. And it produces then a bunch of warnings saying like that, yes, this is the most similar type strain, but it's actually really not that similar to this type strain. It's actually clearly something that's very novel. So, it's sort of a different view of your data that is, very useful if you're mostly interested in comparing things to type strains. All right, so just to conclude here, we started with discussing nomenclature. So this is basically, there's a formal set of rules for assigning names and a committee that basically validates proposals of new names. We then talked about taxonomy and the fact that there is no official taxonomy, just taxonomic opinion. And then we discussed basically species definitions. So how can, in a practical sense, can we assign strains to a species? And in particular, we talked about the polyphasic approach. And then I basically introduced this idea that in a practical sense, sometimes it's much easier just to focus in on the genomic similarity of genomes. So we have sort of a genomic or ANI-based approach, which is what's done in the GTB. And we talked about the GTB in some detail. So this is a genome-based taxonomy, which puts forth some quantitative sort of guidelines for delineating species and higher taxa, and is basically meant to address the current situation where we are discovering new species and new taxa at a very rapid rate. So we'd like some taxonomic framework that allows us to discuss that diversity. All right, so that concludes week 7. Next week, you'll be with Ben Bolduc. I believe he's doing the next 2 weeks, and we'll be switching gears over to viruses. Next week, I believe he's focusing on the tools that are available at KBase and JGI. I wanted to acknowledge that the GTVB is, of course, the efforts of a number of people, all of which are depicted here. It's been a great group of people to work with, so all the thanks to them. And thank you for your time, and I'm happy to take any questions that there may be. Is there a formal way that we're doing questions? Are the moderators compiling those?
Are you able to see the Q&A window?
Okay, is the idea here that I will just go through each of these that are in this window?
Sure.
Okay, so the first question here is just for clarity. By strain, we are also referring to analysis of the whole genome of the particular species and mapping all the pathways and genes it comprises of, correct? Yeah, so I mean, I think strain is a biological concept. So certainly in the context of the GTVB, we're talking about having either isolate genomes or MAGs. So, that is not always the full genome. MAGs are often partial. And people do use the concept of strains even when they have only a very small subset of the available genome. So we can of course still talk about strains even if we're just using sort of 16S sequences. The next question here, can you recommend a database that has an updated cyanobacteria taxonomy? So, I mean, we do have, of course, we classify the cyanobacteria in the GTVB. I know that Phil and Marcia, so they're 2 of the GTV curators, and Russell Sue are working with other people in the cyanobacterial community to put forth, they're working on a new manuscript that's going to do some reclassification, but my understanding that's still a work in progress. Next question here, to any extent this paper is useful for the context of the day. Okay, I will direct people to that. Okay, I'll move to the next question here by Andreas. How can this so I'm going to assume, so the question is, how can this plots be made? So I'm assuming those are referring to the relative evolutionary divergence plots that I showed. That's a great question. There is a program that I wrote called phylorank, which will calculate relative evolutionary divergence on a tree and will output a plot exactly as I showed there, a phylorank. We actually internally refer to those as phylorank plots. Now you do need to provide a tree that is decorated. So the tree needs to, on any internal node that you want to define a taxa, that needs to be defined in your actual Newick tree that you provide to phylorank. So I'll skip down to this anonymous question here. I think sometimes I've heard that people use the concept of variant and strain undistinguishably. Is this correct? Genomically, how can we distinguish variants from strains in bacteria? Yeah, excellent question. I'm sure opinions on this vary a lot. So, I come from a very technical background, and I think that the reality, as I see it, is most of these terms don't have strict technical definitions. So we use the term strain a lot, and we also use the term variant. But exactly what those means is essentially up to interpretation. So I don't think, there is no strict definition to these things. So, it's basically a matter of taxonomic opinion of whether or not you believe, well, I don't know if that's true. So yeah, I think I'll just leave it there to say that, these things don't really have very strict definitions. So there's sort of room for interpretation there. The next question here is, digital DNA hybridization a valid parameter for genome taxonomy, or ANI should be preferred? Yeah, so there are a number of different methods that have been put forth for measuring the similarity of genomes. I do think ANI is the most sort of widely used in our community, but certainly digital DNA hybridization, it's used, for example, at the type strain genome server. I think it's a perfectly valid measure, and certainly a lot of people are recommending the use of sort of multiple of these measures in order to decide if a strain should be assigned to a genome. So yeah, certainly completely valid. So it's just really a matter of, as a community, what criteria we want to use for sort of resolving these questions. All right, next question here. Do you recommend using the GTB16S sequences for building a CHIME database, or no, because the taxonomy is not built based on the 16S? Yeah, great question. So there are, I mean, you certainly can do that. That's the, but there are some limitations you should be aware of. So the 16S sequences at GTB, we take those straight from the genome, and right now we're actually not doing any quality control on those. So that would be something that's left to the users of the GTB. And that is, it doesn't happen often, but it definitely does happen that you have mags that have a 16S sequence in them that is contamination. So we know that happens. And we are working on a method to resolve that, but that's still in development. So that is something to keep in mind. I don't think it matters that the taxonomy is not based on 16S, but it certainly is the case that if you infer a 16S tree, there are likely going to be some GTB taxa that end up being polyphyletic in that 16S tree. So whether or not that matters, I think it depends on the exact application of how you're using those 16S sequences. What I also can say is that, Silva, I think, is, the most widely used 16S resource, and they have started to incorporate, proposals in the GTB taxonomy into the Silva classification when those sort of when the GTB taxa are a better fit to their 16S tree than what they're seeing at NCBI. All right, so next question here. Say you have different gene sizes with multiple functional domains on the same operon, and some sections of these functional domains have been predicted and vary significantly in length. Some very long, some short, either just the N-terminal or C-terminal. What guides the GTB-TK trimming of concatenated proteins alignments of these sequences to assign accurate taxonomy of the data? So mostly it comes down to the consistency in which we can detect the genes that are used to infer the actual GTB reference trees. So for bacteria, we use 120 genes, and these are genes that are largely ubiquitous. So they're found in, I believe, more than 90% of the species we had when we originally put together that marker set. And they're identified using an actual HMM model, right? So that we know that that gene matches this model and can be aligned using that specific HMM model, which essentially wouldn't happen if, say, there was two domains got shifted around, it would no longer match the model. So it's pretty much relying on the fact that those genes are ubiquitous and can be put into a common multiple sequence alignment space, that we know that there hasn't been domain shifting within multi-protein domains. Now, it is, of course, well, and PFAMs, their HMMs are actually at the domain level. So that also addresses that for those subset of genes. Tiger FAMs, as you sort of implying here, are at the full gene level. But our ability to align them and detect them across a bunch of genes is gives us, well, basically ensures that there's not this domain shifting happening. All right, I'm happy to take any other questions if people want to fire them into the chat. All right, if there's no more questions, thank you everybody for your time, and yeah, enjoy the rest of your day. Thank you.